Our next two speakers are YY An and Sebastian Annert, and they will be presenting some work that they did when they were working with Laszlo Barabasi at Northeastern University in Boston on finding the networks of flavors through sort of like different countries. Right now, each of them is in different places. So YY An is assistant professor in Indiana University in computer science, and Sebastian is a research fellow in the Theory of Condensed Matter group in the Department of Physics in the University of Cambridge. So please join me in welcoming them.
Hi, my name is Yongnyeol An, but you can call me YY, which is much easier. And I'll talk about flavor network and the principle of food pairing. So this work is with Sebastian here, and James Bagro, and Laszlo Barabasi. So yeah, I put warning here, but I think Marx shows many nice pictures. Yeah, never mind. So there is really famous saying about food. So tell me what to eat, and I will tell you what to are, right? And it's true in many cases. For example, if you look at pandas, they eat bamboo trees, and that defines their life. I mean, panda has even additional thumb here to grab the bamboo properly, and panda even lost their ability to appreciate the meat, the glutamate. And then we can think about what do we eat. We eat those kind of things. Maybe not here, maybe in US. So if you look at this hamburger, it contains quite a lot of diverse ingredients. For example, meat, bread, vegetables, fruit, cheese, dairy, so many things. And this is one example of Korean dish, full dish, and it has so many ingredients in them. So basically, what I want to say is we are the omnivores, which means we eat everything. And there was a famous book by Michael Pollan, which is called Omnivore's Dilemma. And the dilemma is the following. So we can eat anything. That means we can choose what to eat for every meal. And that's pretty hard compared with this guy, because there are many dangerous things out there, like poisonous mushrooms or fish. Yeah, we should choose those healthy food from all different possible potential food out there. And we have nice metabolic network inside our cells, so they can convert some of ingredient into another, but it has limitation, and we should carefully choose our food. So the question is, how do we choose our food? And if you eat something, why do we eat this? Right? And you may answer that because it's delicious, right? But let's think about why is it delicious? Why is it delicious?
Tastes good.
Eh, Sorry? Sugar? Yeah, that's an important point, because energy is the primary reason that we eat food, right? We need energy to function, and that's why basically the most primary reason to eat food. And that's why if you have a sweetness and fattiness, and that just become awesome, right? Like everybody loves ice cream and cheesecake, right? So energy is pretty important. And even the newborn who had no idea about the real food, they can distinguish the sweet food with like sour or spicy or horrible food, right? But there is another interesting question, which is, then why do we eat spices? Why do we eat spices?
Safety, for safety reasons.
Safety?
Circulation.
I think the most convincing answer is safety. There was a nice paper called Darwinian Gastronomy, and which basically shows that if you plot the temperature versus use of spices, then you have a clear positive correlation between temperature and use of spices. So Thailand, India, they are the most spicy countries, and the Scandinavian countries are least spicy countries. So we know many principles that governs our taste for food. Our question is, are there any other principle that transcend individuals and cultures? That's the question. And we focused on the food pairing, because in almost every meal we put together some different ingredient, right? So there was a famous hypothesis called food pairing hypothesis by François Benji and Heston Blumenthal that says two ingredients taste good together if they share flavor compound. So yeah, it sounds nice. You can generate many interesting combinations from this, but we are scientists, so we are thinking, oh, is it really true or just anecdotal, like fluke? So we wanted to do a little bit systematic approach for this. So basically, we got some data of flavor compound and ingredient relationship, and we got some data here, and we made the network between ingredient and compound. So the left side is our flavoring culinary ingredient, and right side are the compound. And then basically from this, you can generate the network between culinary ingredient. Here the edges, the links, are made if they share many flavor compound. So for example, Parmesan cheese and mozzarella cheese share many compound because they are both cheese. So we can make this huge network, and that's too dense because most ingredients share compound. So we apply some network algorithm to reduce this into more compact or more informative form. That's the figure that appeared in the previous talk. So basically here, the nodes, the circles are ingredient, and they're connected if they share a lot of flavor compound, like statistically significant number of flavor compound. So, and the size of the circles represent the occurrences in our recipes. For example, cheese is close to like wines, and wines are close to like fruits. And you can see there are some meat and seafood and some vegetables. And interesting thing is there are several bridges that connect different islands of food groups, and they are basically beer and black tea and coffee. That's kind of interesting from this network. And the question is now, how can we know about the people's preference on the food? And the answer, our answer is recipes. So we crawled like about 55,000 recipes from 3 online database. And one of them is Korean database, actually, to really control whether there is a cultural bias or not. So basically, it contains recipes from many different regions in the world. And these are the data. And we got a fairly good correlation between different data sets. For example, Korean data set have a fairly good correlation with North American data set in terms of ingredient. And this is the number of ingredient in a recipe. It's about 8 is the largest number, and you have some recipes that have like 30 ingredients. And then you can see like the ingredient frequency, like how much we use this particular ingredient. And butter and egg are there, obviously. So basically, a recipe is a subgraph in the flavor network. Like if you pick some ingredient from the whole network, then it becomes a recipe. So we a little bit generalize this food pairing hypothesis as like this. So are these subgraphs really denser than random subgraphs? So that means we play the surreal food game. So we generate random recipes by controlling some of the aspects of the recipes, and then we compare it with the real recipes and see how they are denser or sparser in real world. So the answer is yes and no. Actually, it turns out that there is a big difference between different cuisines. For example, North American and Western European food, they tend to share more flavor compound in their recipes, while East Asian recipes share much less than random. So they tend to put different ingredients together. And you can see correlation, nice correlation here. And it's very consistent throughout the 3 data sets. And I will skip detail here. We can actually calculate which ingredient is responsible for this effect. And the other one is the idea of flavor principles, which is, let's say, what are the most authentic ingredients of a given cuisine? That's the idea of a flavor principle, which suggested in 1960s. And basically, we can automatically calculate the authenticity of ingredient in each cuisine. For example, East Asian cuisine have many green authentic ingredient, while North American has very white and brown, which consists with Mark's presentation. And we can draw kind of map between different cuisines, but based on this authentic ingredient. So as a summary, we built the flavor network, and I think we should have known Mark before doing this, because he has so many nice data and he's been working on this. So I think we should collaborate. And there is a striking difference between Western and Eastern cuisine. And I think that's kind of interesting. And as reiterating Mark's point, we can do really nice things with like quantized data of food. And we can develop culinary informatics. And as a final thing, there is a nice comic of Garfield. And he asks, isn't it a shame there's no unit of measure for how good food tastes? But Garfield says, oh, there is. Can you guess what is the unit?
The plate's clean.
No. Yeah, it's called calorie. Okay, then Sebastian will talk about more big picture and future work of our research.
Okay, thanks. It's not actually on.
No, it's okay.
Great, thanks. Yeah, I'm going to put this project we did a bit more into a bigger context, and also, I'm going to present a few additional results we've had recently to add to the picture. So yes, I'm in the physics department at the University of Cambridge, and but I spent 6 months in the same group as YY at Northeastern, which is where we started collaborating on this. So over the past 20 years, biology as a science has undergone a huge revolution. And you all know we can now read genomes. We can read the genomes of all kinds of plants and animals. And the cost of genome sequencing is rapidly going down. So in sort of 20 years' time, it will be very feasible to sequence any genome, at a tiny cost and in very short time. We can also, at the moment, already measure the expression levels of thousands of genes simultaneously. What that means is we can measure whether genes switch on and off at the same time in an organism. We can do that in what's called high throughput experiments. So we can make many such measurements. So we can really track how genes influence each other. We biologists also have huge amounts of data on structural data on proteins. That means they have the complete atomic structures of thousands and thousands of proteins and protein complexes, which are even bigger structures. And why am I telling you this? Well, it's basically the upshot is that biologists have access and have to deal with enormous quantities of data. And this is something that has changed a lot over the last 10, 20 years. So the whole field of biology has to evolve. And to analyze this data, biologists had to essentially invent a whole new field called bioinformatics, which invents new methods to look at huge data sets, to extract biological meaning from them. And it's a huge industry. It's thousands and thousands of researchers working on it all over the world. And it's a very new phenomenon. And similarly, computational biology is sort of a related field, a bit more inside biology, where you use computing power to predict biological structure of proteins or the genome sequences. And biologists also had to come up with a sort of new way of looking at biology, and that's called systems biology. So you try and look at the system as a whole. You try and, before biologists would look at a very small, a very detailed mechanism and study that for a very long time, just because it's so complex. But now they can sort of step back and look at the whole picture in a whole new way by making use of the collective data sets. And they also employ theoretical models to try and predict what they're going to find. In other words, systems biology tries to derive fundamental principles by looking at the system and the data as a whole. And now you might ask, what does this have to do with food and gastronomy? Well, this is not isolated to biology. This is actually a broader trend in science and even the arts and humanities at the moment, that you get similar kind of data revolutions taking place in other areas. And one really nice example is language. So we all know about Google Books and about similar efforts where you have millions of texts online. And but this, in fact, can lead to the new discovery of new and fundamental laws about language, about the evolution of language. Essentially what linguists used to do, and still do, obviously, but it used to be the exclusive preserve of linguists. Whereas now, there was a paper in Nature a few years ago which found this relationship, which might look a bit complex at first. It's the half-life of an irregular verb scales as the square root of its usage frequency. And what that means is, basically, you've got regular verbs and irregular verbs. So verbs that, for instance, to help has the past tense helped. But actually, if you look at Old English, it was actually holp. So it was a different kind of past tense. It was a strong verb rather than a weak verb. And what these people found is that if a verb gets used 100 times less in language over the course of time, it regularizes 10 times faster. That means it becomes a regular verb at a much quicker rate if it gets used less. And while you might have guessed that conceptually or qualitatively, these guys managed to put a quantitative relationship on this. And they did that purely by analyzing many, many texts digitally. And interestingly, the authors of this paper are not sort of traditional linguists. They're in the departments of evolutionary biology, applied maths, and systems biology. So we really get this, this is sort of part of the bigger trend that we look at huge data sets in a quantitative way and try to derive fundamental laws from them. And so YY has already shown you our work on the flavor network. And this is a perfect example of what we're thinking of is to bring the methods of this systems biology, of large-scale data analysis, to food and gastronomy in a very broad sense. And so what YY, the project YY talked about, we based on flavor compounds and recipe data sets. Now, to test the shared compound hypothesis that foods that share compounds are more likely to taste well together. Now, we found some support for this hypothesis, but since we finished this project, we've been thinking more about it. And what we realized when working with recipe data sets is that they're incredibly complex. They're very multi-layered, and that's what came out of Mark's presentation, of course, as well. And I really liked your tree, the way you broke down recipes into trees. And maybe one could work with that more. But we quite struggled with the complexity of recipes. And because they have information on ingredients, relative quantities, cooking techniques, but also, importantly, recipe ingredients are not just about flavor. They're a lot about texture and about the structure of the food, of the dish you're making. So not everything is about flavor in recipes. Can we somehow get a clearer picture by using other types of data, was our question. And yes, because we got hold of a curated list of flavor pairings. So this is where chefs have gone out and well, actually, a pair of authors have gone out and talked to chefs and collected a long list of pairs of flavors that go well together. And they even have a sort of strength ranking or sort of strength of recommendation level as well. And you might have heard of it. It's a book, published as a book called The Flavor Bible by Karen Page and Andrew Donenberg. And very nice to just read through and get some ideas for your own cooking as well. But so we used this data. And we also got a new data set on, so in the, 5 minutes. In the YY's talk, he talked about Fenaroli, which is a flavor handbook where we got our original data from. This was, we now have access to the VCF database, which is a commercial database of food ingredients and the flavor compounds they contain. And it's got the advantage, it's also got for a lot of the compounds, it's got the concentrations as well. So we know how much of each compound roughly is in each ingredient. Now this is good because it allows us to distinguish 100 parts per million from 1 part per million, and therefore to say that something that's present at a level of 100 parts per million is more likely to have an influence. What we'd ideally odor activity values, which take into account that some compounds we can taste much more easily than other compounds. So it doesn't take into account our relative taste sensitivity, which would be kind of the next level. But at least we've got the relative concentration, so that's already a step forward. And we also make use of a new kind of data, which is description. So flavor compounds all have a description by a flavor chemist who sniffs the liquid and says that smells like strawberry, and he writes that down, and that's an official, reasonably official flavor description. So we use that data as well. And now we've got descriptions, the flavor Bible, and the VCF database. Can we get something new out of this? Can we add to what YY has already said? And the result is encapsulated in this figure, which I'll just quickly explain. You see here from left to right, we are increasing the concentration threshold. So we only consider ingredient-compound relationships above, say, 10 parts per million, 20, 50, 100 parts per million. This is a significance measure. So this is Z-score, which is the normalized. So this is standard deviations between the data and a randomized control. So sort of biologists would usually be happy with about 2 standard deviations as something biologically significant, something they can publish. The 3 colors are, we consider all compounds with a description only, or compounds with descriptions that contain food items. So this is basically compounds that smell or taste of food. And if we do that, then we find an interesting picture, namely that once we start ignoring the smallest compounds, or the sort of weakest, least significant compounds, and if we only look at compounds that smell or taste of food, we get quite a significant increase, quite a significantly more shared compounds in the flavor Bible pairs than you would expect by chance. So what that means is that the pairings in the flavor Bible share more compounds than you would expect by chance. And this goes particularly, this goes particularly for if you only consider compounds that smell or taste of food. So just to summarize, this is the sort of original hypothesis we started out with, and we found some evidence for that. But if you look a bit closer with this new data, it seems more like if food share dominant flavor compounds that have food aromas, they're more likely to taste good together. So it's kind of a refined version of the original hypothesis. And the even bigger conclusion to the whole talk is that culinary science really can benefit from large-scale data analysis. And I think we've already mentioned culinary informatics. And I think gastronomy is really heading that way, or the kitchen science really could benefit from heading that way for some new fundamental discoveries and new ways of looking at food. So my collaborators were already mentioned in Waiwai's talk. I just mentioned them again. James Bagru, who's now at Northwestern, and Laszlo Barabasi, who's still at Northeastern. And it just remains to thank Ben Nyssen and his colleagues at TNO for giving us access to the VCF data for research purposes. And finally, I'd like to thank you for your attention.
Thank you. Do you have any quick questions for Wai or Sebastian? Anyone has any question? You have a question? There's a question here. Here, the gentleman in the white shirt.
Good morning. Did you consider the crunchiness as a factor? The crunchiness as a factor to hook it, your taste, your
crunchiness, texture.
Sorry?
He says,
Did you consider texture?
I mean crunchy, the crunchiness.
Being crunchy.
Crunchiness.
The crunchiness of the food, if that was one of the elements that you took into account. Is it part of a flavor, being crunchy?
Oh, no. That would be that's what we're trying to separate out in the analysis. So in the original, in the recipe analysis, that would have come in. But what we tried to do in this recent work is to try and separate just out the flavors. So texture didn't come into this the second analysis. Oh, yeah.
Is there any other question? I have a question related to the last study versus the original study. So on what you presented, why you showed that for Eastern cuisine, the flavor compound hypothesis didn't seem to hold because they were mixing foods with different compounds. But in the latest results, you seem to have put all of the cuisines together.
Well,
so
it's a very different data set. So in the flavor Bible is not arranged by cuisines. So it's just a cross cuisine. It's just chefs saying what they would put together. It's not, so we don't have any, with the recipes, we've got an automatic categorization by cuisines. So what might be interesting is to look at
The bias on the
to try and classify the flavor
Yeah.
Bible, and then to check whether that bias is still there, or whether that bias is maybe something to do with the complexity of recipes. Yeah.
Okay. Okay. Thank you. Well, let's thank you, the speakers.